Right now, inside almost every cell of your body, there is something invisible, but incredibly precise. A structure made of approximately 3.2 billion DNA base pairs. This is called the human genome. And it is not random. It is arranged in a specific biological sequence that defines how your body is built, from the color of your eyes to the way your immune system responds to viruses. If you were to write out just one human genome in plain text, it would take up over 3 million pages. That is roughly the equivalent of thousands of books stored inside something smaller than the width of a human hair. And yet, all of this information is packed inside the nucleus of a single cell. A structure so small that you cannot see it without a microscope. But here is something even more difficult to imagine. You are not made of one genome. You are made of trillions of cells, and almost every single one of them carries this same instruction set. The same 3.2 billion letter code, repeated across your entire body. Now the question becomes more interesting. If we have had access to the human genome for decades, why don't we fully understand it? The Human Genome Project officially mapped the sequence of human DNA in 2003. It was considered one of the greatest scientific achievements in history. But even after mapping it, scientists discovered something unexpected. Knowing the sequence is not the same as understanding its function, because inside that sequence, there are regions that do not behave like traditional genes. Large portions of DNA do not directly code for proteins, the molecules that build your body. At first, scientists called this non-coding DNA. Later, it was even labeled as junk DNA. But the more we studied it, the less accurate that explanation became. Because these regions are not inactive. They appear to play roles in regulating when genes turn on and off, how cells specialize, and how complex biological systems are coordinated. In other words, we know the language of DNA, but we do not fully understand the grammar. And without grammar, meaning becomes incomplete. So even now, with all modern science, we are still facing a simple but unsettling reality. We have mapped the code of life, but we have not yet fully decoded its meaning. What DNA actually does, so what exactly is this system doing inside you? DNA is built from 4 chemical bases, A, T, C, and G. These molecules pair in a very specific way. A always pairs with T, and C always pairs with G. This pairing forms the famous structure known as the double helix. It was first described in 1953 by James Watson and Francis Crick based on X-ray images produced by Rosalind Franklin, one of the key contributors whose role was only fully recognized decades later. But the important thing is this. DNA itself is not active in the sense of building your body directly. It acts more like a stored instruction system. And to make those instructions usable, your cells go through a multi-step process, DNA to RNA to proteins. This process is called gene expression. First, a section of DNA is copied into RNA, a molecule that acts like a temporary working version of the instruction. Then, this RNA is used to build proteins. And proteins are where biology actually becomes physical reality. Proteins are responsible for forming muscle fibers that allow movement, building enzymes that control chemical reactions in your body, creating hemoglobin that carries oxygen in your blood, regulating neurotransmitters that affect mood and thinking, and controlling immune system responses to infections. In fact, nearly every biological process you can think of depends on proteins. Now here is where things become interesting. The human genome contains around 20,000 protein-coding genes. That might sound like a lot, but compared to other organisms, it is not. Some plants have more genes than humans. Some amphibians also exceed us in gene count, so complexity is not just about number of genes, it is about how they are controlled. And here is the real shock: those 20,000 genes make up only about 1 to 2% of your entire DNA. The remaining 98 to 99% does not directly code for proteins. For a long time, scientists did not know what to do with this information. In the early years of genetics, especially before the 2000s, this non-coding region was dismissed as evolutionary leftovers. They called it junk DNA. But that term is now considered outdated, because when large-scale projects like the Human Genome Project, completed in 2003, and later research initiatives began analyzing these regions in detail, something unexpected appeared. These non-coding regions are not empty. Many of them contain regulatory sequences that control gene activation, enhancers that increase or decrease gene expression, silencers that shut genes off completely, and structural regions that help organize DNA inside the nucleus. In other words, they do not build proteins directly, but they control when, where, and how proteins are built. And that changes everything, because it means DNA is not just a static instruction list. It is a dynamic regulatory system. A system that decides which parts of your body are active at any moment in time. And even more surprisingly, a single gene can be influenced by multiple regulatory elements located far away on the DNA strand, sometimes thousands or even millions of base pairs apart. So instead of a simple linear code, what we actually have is closer to a three-dimensional control network inside the cell nucleus. And this leads to a deeper scientific problem. We know how to read the letters of DNA. We can sequence it. We can map it. But understanding how all these regulatory layers interact in real biological time is still one of the biggest challenges in modern biology. So even after decoding the human genome, a fundamental question remains open. If only 1 to 2% of DNA builds the body, what exactly is the remaining 98% actually coordinating? Beyond Human is just getting started. Subscribe to witness what comes next.